Welcome to part 1 of 2 for our lecture that covers chapter 8 from your textbook on experimental research. So as all the chapters do, this chapter begins with a discussion of existing research, and specifically it's looking at this trending topic of commercially based products that are often called cognitive or brain training programs that are purported to improve mental functioning. I work a lot with patients with dementia or with mild cognitive impairment that has not quite led to or is not going to lead to dementia. And a lot of them ask me, what can I do to improve my mental functioning? Are there any exercises or things I can do? So there's this common conception that people have that, you can essentially build up cognitive muscles the same way we do physical ones, and that perhaps some of these programs can be employed to stave off or to slow down the progression of various types of cognitive deficits. So the question that's really posed in the chapter is what kind of study and what kind of evidence would be needed to determine whether or not these computerized cognitive training programs work, if they do in fact improve mental functioning. So the research obviously implies the idea of cause and effect. In the statement itself, there's this assumption of cause and effect. So the hypothesis that's to be tested seeks to predict whether or not, to predict that cognitive training leads to improved mental functioning, basically. So the hypothesis dictates a specific type of research design known as an experimental design, in this case a randomized true experiment. So there's research that's done by Merzenich and colleagues that's described throughout the chapter. They looked at the concept of brain plasticity. The idea is the brain pliable? Can it be exercised or developed in various ways? And the chapter begins by examining experimental design's critical and defining features and how they provide the strongest test of a causal hypothesis. This is the thing we've referred to as sometimes the gold standard, right? It is a true experiment that this really gives us the best amount of internal validity, the best ability to make a cause and effect argument. So the question then is, can you essentially do mental calisthenics for your brain? And we're going to talk about a true experiment that tries to address this issue. So in a true experiment, the hypothesized cause is the independent variable, this thing that's manipulated by the researchers, and the dependent variable, the outcome, is the hypothesized effect. So Merzenich and his colleagues developed a brain plasticity study that examines the theory that exercising your brain via a very specific type of memory recall improves overall mental functioning. So exercising the brain is the independent variable, and overall mental functioning is the dependent variable, or the outcome they're interested in. They designed a true experiment which randomly assigned, that's an important aspect of an experiment, remember, randomly assigned participants to either one of three groups. Okay, so there is the first group, which you might call the experimental group. This is the brain plasticity computerized training. Participants in this condition of the independent variable receive the brain plasticity training that's hypothesized to improve mental functioning. There's a condition called an active computer-based control, which is where the participants engage in a similar amount of face time with a computerized program, but it is not related to brain plasticity or cognitive functioning at all. It's simply taking the place of something being done. It's a control condition. It's trying to control essentially for the placebo effect. And then finally, there's a third condition, which is a true control called the no contact control, meaning that the people in this group don't undergo any kind of training. So they use this computer-based control condition, as I mentioned, to help ensure that the observed improvement in mental abilities could be attributed to the treatment they were interested in, the brain plasticity treatment, rather than the placebo effect, which is really long confounded cognitive enhancement studies. People believing that they're doing something that's meant to improve their functioning actually causes them to perform better on outcome tests. So experiments are all about causation, and we're going to talk about how we break this down logically. Essentially, we want to focus on what are called the three prongs of causation. So in a scientific sense, explaining something means identifying its cause. By explaining it, you're identifying its cause. So you're establishing a causative agent, and this is sort of the holy grail of scientific explanation and understanding. A true experiment provides a research design to test for whether the manipulation of the independent variable caused the change in the dependent variable, the outcome. So the cause and effect relationship between independent and dependent variables is supported by these three kinds of evidence, these three prongs of causation, as described by Cook and Campbell back in 1979. So essentially, we want to look for these three conditions to make a strong argument for causality. First and foremost, temporal precedence. This is one of the things we discussed in our chapter on correlation when we were doing cross-sectional analyses, when we were gathering data at one point in time with no follow-up, no longitudinal follow-up. And we talked about how there was no temporal precedence. Therefore, you knew these things were related by a strong correlation coefficient, but you didn't know whether or not one caused the other because you were looking at a sliver in time. In an experiment, when we're trying to argue for causation, we want to see that the cause precedes the effect in time. Logically speaking, the cause cannot follow the effect in terms of the timing. The second step is the covariation of the cause and the effect, which is essentially the correlation between the cause and the effect. When the cause is present, is the effect more likely to occur? When the cause is absent, is the effect less likely to occur? This can be thought of logically as if X is the cause and Y is the effect, then if X, then Y, or if not X, then not Y. What we're really just talking about here is correlation. As one changes, as the likelihood of the cause increases, is the likelihood of the effect increasing as well. And then finally, as we've discussed in previous chapters, the elimination of alternative explanations. We want to try and show that nothing else is responsible for this identified relationship between the cause and the effect. If there is no other plausible explanation for that relationship between the proposed cause and effect, if we've got covariation present, usually measured through correlation of some kind, and if we've got temporal precedence, then we can make a general argument about cause and effect. So let's talk a little bit about the basis of a lot of experimental design, and really that gets back to the idea of research and theory. So we know that scientific research is fueled by theories, right? We develop theories about how variables are connected to one another, how they're related, and this is really what not only feeds our understanding of a concept, but also then drives the research, because we want to flesh out the theory, we want to test all the aspects and the relationships that are proposed in the theory. So they're very important in explaining pure empirical findings, and of course we derive our hypotheses from theories. If you go back to our discussion about conceptual and operational definitions, if you remember that conceptual definitions are those that are abstract, they're not existing in the material world. They're concepts like memory or attention, and they can't be pointed to or specifically measured in any way. We have to then develop an operational, quantifiable definition that we can look at in the real world and measure and somehow test. Now, that same process kind of happens with theory and hypotheses. So the theory is more conceptual, the theory is more abstract, but from those theories we formulate and derive our specific hypotheses that we're interested in. I'm going to go into some detail regarding an example here, and this is something called the Stroop test. And it was developed by John Ridley Stroop, and essentially the Stroop test is, and its development is an example of a well-controlled experimental task. And I'm going to define what the Stroop test is in just a moment. There is essentially a way of trying to confuse the ability of the brain to shift sets. And the Stroop test, by basically employing both words and colors, is trying to throw off people's their implicit reactions, their sort of instinctual reactions, and see if they are able to inhibit behavior in various ways. So the Stroop test is really one of those things that seems very simple, and we'll describe it in a moment, but what you find is that it had a really significant role in building and testing psychological theories of cognition in general, and our understanding of how the brain works. So Stroop essentially, in his test, he has a word list where the participant is instructed to say the font color, right, and not to read the word. I'm going to show you these examples in just a moment. You're presented with a list of words, okay, a series of words, and those words are all written in different colors. When the person looks at that list of words, we have a natural inclination, because we are so language-based in our thinking, because we're linguistic creatures, assuming that the person who's doing the test is literate, the idea is that they are very inclined to simply read the word. However, that's not the task at hand. The assignment that you're given in the Stroop test is to name the color of each word sequentially as you go through the list. So the idea is whether or not you can inhibit yourself. Now that's in the sort of one example of the Stroop test, which is called the incongruent example, where the words are the letter, for example, the word yellow is not written in a yellow font. It's written in another color. So that's where you really have to get into this inhibitory control. So the independent variable in this case is essentially manipulating between congruent and incongruent. So in the congruent condition, and which I'll show you examples of both in a moment, the word blue is written in a blue font. In the congruent condition, the word red is written in a red font, so on and so forth. In the incongruent condition, the word red might be written in blue type. The word yellow might be written in red type. So the idea is that the word itself and the color that it's written in are incongruent. They don't match up. What you then can do is you can present both of these lists to the individual, and you can ask them to read the list out loud, and you time them from start to finish. The congruent list is usually presented first, and then the incongruent list. The difference between your response times is really the dependent variable. How much longer it takes you to complete the incongruent list compared to the congruent list, keeping in mind there's the same number of words on each list. So the dependent variable, the outcome you're interested in, is the difference in response time. So Stroop really concentrated on specific key aspects of attention, and specifically this thing called inhibitory control. Inhibitory control is a huge part of how our frontal lobe in our brain functions. We have these very strong, dominant, prepotent responses, and we have a natural inclination to read the words, for example, in the case of the Stroop test. It requires a certain amount of cognitive energy, and believe it or not, some pretty developed ability in the frontal lobe of the brain, some pretty much higher functioning, cerebral functioning aspects of our brains and how they work to inhibit that response and to actually remember the instructions in each instance and say the color clearly. So he's really, in a sense, touching on some very important aspects of how our brains work. If you look at patients with dementia, especially frontal lobe dementia, or frontotemporal dementia, excuse me, what you find is you see really poor inhibition in people with more advanced cases of these types of conditions. They say and do things they wouldn't otherwise do because they are impulsive. They can't inhibit their behavior. They display what's called disinhibition in this case. So he takes this abstract concept of how our brains work and this inhibitory control, very high-level functioning thing, and devises this very elegant, simple way of measuring it and investigating it, which, as I'll show you in a minute, is actually quite cool. And if you do the test, you'll find that there is a difference in your response times between the two lists, and you're certainly welcome to do that as a personal experiment. That inhibitory control, the degree of inhibitory control, is operationalized by Stroop by simply subtracting the response time between, the congruent and the incongruent trials. So this is an example of a study that's designed to evaluate a theory. So this is the congruent Stroop test. Now I'm going to show you not perfectly equal examples here because of just the images that I was able to pull. This actually is written in the front of your book, the front cover of your book, because there's not full color photos in your book. It's written on the inside of the front cover. But as you see, this is the example of the congruent test. So if I were the experimenter, I would present I would explain the instructions to the participant first. I would say, I'm going to show you a list, and I would like you to simply list off the colors that the words are written in, one at a time, sequentially. For example, in the congruent Stroop test, where the colors match the words themselves, I would simply look at this and say blue, red, magenta, yellow, red, black, magenta, so on and so forth, and the person would be timing me and how long it took me. And then we would move on to the incongruent Stroop test. So again, the list is not exactly the same. It will be identical in terms of the number of words included, and this one the font's a little different, but you get the idea, right? In this case, the words are written in different colors. So the word purple is written in red font, for example. The word yellow is written in blue. So now when a person looks at this, they have this powerful dominant response that's part of how our brains work that wants to look at this list and say purple, yellow, red, black, red, green, red, yellow, orange, blue, purple, black, red, green, orange. Okay, that's the linguistic prepotent response that's there. What I have to train my brain to do, or I have to engage in my brain in terms of inhibitory control when I'm in this condition, this incongruent condition, is I have to say red, blue, green, yellow, green, black, blue. It's harder than you think, right? So as you get into it, as you notice, I very quickly slowed down, right? I was having to inhibit my natural response. So this is just one example in your book of a really fascinating way in which we can utilize a simple experimental design, manipulate a variable in a very simple way with two conditions, and end up with the ability to look at how the brain functions and actually come to some pretty significant, well-replicated conclusions about cause and effect within the human brain. So let's talk a little bit about experimental designs and what they look like. We're going to start with what's called a single factor experiment. So a single factor experiment is one in which there is only one independent variable. We sometimes refer to independent variables as factors. You'll see, or you might even see the term multifactorial, but certainly single factor is the implication that there is only one independent variable within this study. So you have complete control within an experiment as the researcher over the manipulation of that independent variable, and in a single factor experiment, there's only one. An experiment with more than one independent variable is often referred to as a multifactorial design. So how do we go about selecting our research participants in an experiment? We first start off, as we discussed in our chapter on samples and populations, we decide who's going to be included and excluded from a sample. For example, there might be some logical, theoretical reasons or practical reasons about who you're going to exclude or include. Stroop chose to present the words only in English in his original test, and therefore non-English speaking people were excluded. As you can imagine, people who are not literate are also excluded from this test. They're not able to participate because of certain characteristics. Exclusion and inclusion requires a lot of care for a variety of reasons. One is not to introduce bias. We also have to avoid certain ethical biases and make sure that we are ensuring adequate representation of the overall population. The ultimate goal, as we've discussed in previous chapters, is to maximize generalization potential to other populations and situations, right? We want to be as inclusive as we can in selecting our sample. We also need an ample number of participants to test the hypothesized effect. This is back to this idea from a previous chapter called a power analysis. This is an understanding about the fact that in most research, the bigger the sample, the better. Okay, because you are essentially getting a larger piece of the pie from the larger population, it's more representative. You're more likely to discover the proposed relationship, the hypothesized relationship, if there is one, with a larger sample. But a power analysis is a way for us to sort of determine how big is big enough in terms of our sample size. What a power analysis does, as we discussed in a previous chapter, is it's a statistical computation where you have an estimate of how strong you think the hypothesized effect is going to be. This is usually based on previous research, but it's also derived a little bit from theory, and can be a place where we can make some subjective mistakes. But it tells us the reasonable number of participants we need within our sample in order to be able to detect this hypothesized effect if it does in fact exist. So as we mentioned in our previous discussions very briefly about experiments, one of the key characteristics of an experiment, of a true experiment or a randomized experiment, is that we randomly assign those research participants. So a true experiment is characterized by this randomization in assigning participants to groups or to different conditions of the independent variables. So if you go back to the brain plasticity study we were discussing at the beginning of this chapter, remember there were three conditions. You were either in the experimental condition that got the brain plasticity training, you were in the computer interaction condition, which was the placebo condition, right? You did something unrelated to brain training, but it took the same amount of time in front of a computer, and then the no contact control. So in this case, we have three levels, 1, 2, 3, of our independent variable. For it to be an experiment, we need to randomly assign our participants to be in one of those three groups. Each individual in our sample has an equal chance of ending up in any of the three groups. There's a concept, there's an idea behind random assignment that I want you to think about. And this is not always sort of patently obvious, which is that people are different, right? They bring something individual. They bring their own degree of error, so to speak, right? They bring their own deviation from the mean with them. And maybe they have a certain characteristic that we are not measuring, that we're unaware of, that really influences the outcome. If I randomly assign people to groups, the hope is that that extraneous influence gets equally distributed across those groups. So by random assignment, I'm trying to ensure that this extraneous influence is just as likely to affect one group as the other, that it's equally distributed. It helps to minimize certain forms of bias, like selection bias, which is a systematic error in sampling, which results in some members of a target population being less likely to be included in the study than others. This is also called sampling bias. So at least random assignment can hopefully help to reduce this. In the Merzenich study we discussed, the brain plasticity study, they randomly assigned participants to one of those three conditions, as I mentioned. This is really in place. This is one of the characteristics of an experiment, of a true experiment, that allows us to demonstrate the cause and effect relationship. If the person comes in and wants to be critical of our findings, they say, well, perhaps there's something about people that influences this outcome. But your answer can then be, because of random assignment, whatever that characteristic is, it was evenly distributed across the three groups. Just by random chance, it should be. Okay, or at least close to. It's never going to be perfect. There's always going to be error. There's always going to be extraneous influence, but you can minimize it and therefore increase the strength of your argument regarding cause and effect. So this is useful for really maximizing the likelihood of what we call internal validity, right? Our ability to make an argument about cause and effect. It doesn't necessarily have a huge influence on generalizability, but it does help with the idea of our ability to talk about cause and effect. So your book uses this term, the theater of independent variables, right? This idea that experiments are really manipulations and design decisions that are made by the researcher to include or exclude certain independent variables, to structure them in various ways so that they increase the likelihood of making a measurable effect found, and therefore making that cause and effect relationship that we're interested in, that hypothesized relationship, be more valid, to be more available to be discovered. So independent variables are created by the researchers, right? They're derived from theory, and they're meant to operationally measure hypothesized constructs out there. But they're really studied in a way and manipulated to determine their impact on the dependent variable. They are the cause, and the effect is measured in the dependent variable, the outcome. As you can imagine, an independent variable must have at least two levels. You have to have at least two conditions to compare to one another. So 2 is the minimum, but of course there can be more. In the Merzenich study, there's 3. The levels of the independent variable need to be truly distinct from one another. Okay, so that you don't have overlap or bleed, that there's not too much overlap, because then you're less likely to find that distinction. You want them to be distinct so that their effect on the independent variable can be objectively assessed and then clearly interpreted through our statistical analysis. There's a way of talking about this relationship between the independent variable and its causal relationship with the dependent variable that's really important as far as understanding statistics and as far as understanding the real principles behind a true experiment. We refer to this condition the null effect, right? Null simply being, a Latin-derived word for none or absence of. So a null effect occurs when the independent variable fails to produce any effect on the dependent variable. Excuse me. Let me repeat that. So the null effect is when the independent variable, manipulation of that independent variable, fails to produce any significant effect on the dependent variable. This is kind of tied to this issue we've discussed much further back in this class about falsifiability. That a hypothesis, in order to be valid, must be falsifiable. That's a foundation of science. The null effect is essentially a falsified result. It's that there is no relationship between the independent variable and the dependent variable. It does not produce this causal effect on the dependent variable by manipulating the independent variable. So the null effect is used to describe negative research findings, meaning that you failed to demonstrate statistical significance. Now that's an important distinction, but there was not enough statistical evidence to make the argument of a cause and effect relationship between the independent and the dependent variable, and we refer to that as the null effect, as a null effect. For example, in a treatment study, failing to show any beneficial effect for the treatment or the intervention. In the Merzenich study, it would be seeing that all 3 of the groups look the same, that whether you got the brain plasticity training, you got this computer control, or you got the no contact control, but there was no real difference in outcome. We would refer to that as the null effect. So we'll pick up there in part 2.